The first event of neurulation is the formation of a thickened area of cells, called the neural plate. The neural plate forms at the cranial end of the embryo and grows in a cranial to caudal direction. The cranial or head end of the neural plate indicates the region of the future brain, and the narrower caudal or tail end represents the future region of the spinal cord. By the end of the third week of development, the lateral edges of the neural plate become elevated and move together to form the neural folds. The resulting space, created by the folding of the neural plate, is called the neural groove. The neural folds fuse together, and the neural plate transforms into the neural tube, the precursor to the central nervous system. Fusion of the neural tube usually begins in the middle of the embryo, extending in both cranial and caudal directions. During the closure of the neural tube, cells on the crest of the neural folds detach, forming a new cell population called the neural crest. These cells contribute to the formation of the peripheral nervous system. Once the neural tube has completely fused, the process of neurulation is complete. During the fourth week of development, a period of rapid growth, the embryo begins to change shape from a flat trilaminar disc into a cylinder, a process known as embryonic folding. Embryonic folding occurs in two planes, the horizontal plane and the median plane, and is the result of differing rates of growth of the embryonic structures. Folding of the embryo in the horizontal plane results in the development of two lateral body folds. Folding in the median plane results in the development of the cranial and caudal folds. Folding in both of these planes takes place simultaneously, resulting in the rapid development of the embryo. The cylinder consists of three layers derived from the trilaminar embryonic disc. These are the endoderm, the innermost layer, the ectoderm, the outermost layer, and the mesoderm, located in between. The endoderm of the trilaminar disc is mainly responsible for the formation of the gastrointestinal tract. As embryonic folding continues, the endoderm moves towards the midline and fuses, incorporating the dorsal part of the yolk sac to create the primitive gut tube. The primitive gut tube differentiates into three main parts: the foregut, midgut, and hindgut. The foregut can be seen at the cranial or head end of the embryo. It is temporarily closed by the oropharyngeal membrane, which at the end of the fourth week of development ruptures to form the mouth. The midgut lies between the fore and hindgut and remains connected to the yolk sac until the fifth week of development. As embryonic folding continues, the connection to the yolk sac narrows into a stalk known as the vitellin duct. The hindgut lies at the caudal or tail end of the embryo. It is temporarily closed by the cloacal membrane, which during the seventh week of development ruptures to form the urogenital and anal openings. As a result of embryonic folding, the major body plan is established, and the three germ layers continue to differentiate, giving rise to their own specific tissues and organ systems.